All right, so I understand there was a Blackboard issue yesterday. Hopefully, it looks like that's been resolved now. So if you had trouble accessing the videos, hopefully that should be okay now. I think there's a couple different issues that looks like they've got resolved. So that's good news that it wasn't my fault. We couldn't get into it. So remember exam the next time we meet on Tuesday. I think I'm gonna have you just come into the room like normal. And I'll pass the exams out. We should have plenty of time to do the exam. I'll start a little bit before the hour. That way we're not all bunching up before we come in. So we'll come in, spread out. I've heard from a few of you that have some special circumstances on Tuesday. I'm really trying to get as many of you to take it in person as possible. So don't take it the wrong way if I'm trying to get you to come in early or maybe take it the day after. But I just think that's important just to kind of have everybody do it the same as much as possible. So let me know if something's up. If you're quarantined, we'll work something out, but let me know what you have going on. Okay, so I'm planning on the exam covering through chapter 14, so we should be able to get through that today unless I really go off tangent too much, but so that's what I'm planning on. All right, so we ended last time beginning the pathogens journey into the host body, so that always starts at a portal of entry. We talked a little bit why the skin is not a good portal of entry. Not a very common one anyway, as long as that skin is intact. Mucous membranes are typically the most common portal of entry. We have defenses. We're going to talk about that as we get into our immunology chapters next week and beyond. We have defenses there, but they're just not quite as good. So we have a thinner layer of cells. Respiratory tract is a big one where we just seem to be a little bit more, again, not have such good defenses there. So that's the most common one. Placenta, there are a few organisms that can cross that placental barrier. The virus that causes German measles is one of those, but that's not very common. The one that's not as obvious, we have a special name for it, the parenteral route. Okay, so not parental, parenteral. If you break that root apart, enteral means intestinal. You'll see that with a few terms that we have coming up. We're going to talk about in the lab this whole group of organisms called the enterics. Okay, these are organisms that are typically found in your intestinal tract. So that enteral means intestinal, and par, that prefix means not or against. So not in the intestinal tract. So a portal of entry that's considered parenteral means not by the normal methods, not by just swallowing something. So this is when a host might have a break in the skin or some kind of puncture wound, or maybe a hypodermic needle that got left off there, an insect bite. So something kind of out of the ordinary has happened that's given the pathogen an opportunity to gain access to our body. Okay. All right, and we talked about the skin. All right, so once a pathogen has access to the host, first thing they have to do is be able to attach to the cells. Okay, so remember, they have to come in by the correct portal of entry where their cells are that they are going to recognize and be able to attach to. Okay, so usually pathogens will have some kind of strategy in order to be able to do that. It might be a physical structure like pili or Fimbriae. Typically, the Fimbriae is what we would consider as an attachment mechanism. Remember, those two structures are made out of the same protein, pilin. So sometimes those two structures, we kind of use those terms a little bit interchangeably. Let me skip to this picture quickly or briefly. Remember, typically, if there's a lot of these short appendages around the cell, we would consider that Fimbriae. Okay. Of course, they're much shorter than flagella. So those function in attachment. Oftentimes, they're going to have some kind of, at least for bacteria, some bacterial attachment proteins. Okay. And I was gonna mention that protozoa often might have another kind of attachment. Tapeworms have that. If you ever seen a blow up of that tapeworm head, they have a scolex, which basically clamps on to the inside of our intestine. So that would be an example of an adhesion factor, maybe. Okay, but typically at the end of that adhesion factor, like the Fimbriae, there's also going to be some proteins there that are specific. So the Fimbriae are going to attach to something specific. And those attachment proteins, often we refer to them as ligands, or sometimes we call them adhesins. Adhesins is a little bit easier to understand what that means, but you'll see the term ligand as well. Okay, these are just proteins that the bacteria has that are going to recognize some specific host structure. Okay, so example, or the graphic from your book here, sorry, some of my headings are chopped off here. I thought we could label this together. This is figure 14.5. Okay, so here's our bacterial cell, and these structures all around the outside of the cell, these are the ligands, or again, sometimes called adhesins. Those are proteins. Now, these are drawn way bigger than what they would actually be in a cell, but just so that you can see them. Okay, and those ligands are going to be recognizing some host cell receptor. Okay, so here's our host cell, here's the plasma membrane here. That host cell receptor can be, it could be a sugar, it could be a protein, it could be a lipid, it could be a combination of any of those. So we're not going to worry about the composition there. The part B here shows some E. coli. This gray part around the E. coli is the capsule that the E. coli produces. So a capsule can help a cell adhere to certain structures as well. This is a transmission electron micrograph, but what you can't really see here, there's going to be adhesin proteins there as well. Okay, so that are going to be a little bit more specific in this case for the membrane of the intestinal wall. Okay, and here's another example. I think I put this picture up with the other one just to kind of conserve paper there. But some pathogens might produce more than one adhesin. This is an example of a pathogen here. This is Bordetella pertussis. That's the organism that causes whooping cough. And it's so important for that organism to adhere to our ciliated epithelial cells. Okay, these are cilia here in our respiratory tract. They have 8 or 9 different toxins or adhesin proteins that help them to adhere to those cells, because they don't actually invade into the cell most of the time. Okay, so it's really important for them to be able to stick there in order to start reproducing and making their toxins. All right, so once that pathogen is sticking to the appropriate kind of cell, most of them will try to invade deeper into the host tissue. So we're going to talk about some different kinds of virulence factors that, remember, virulence factors are tools that an organism would have in order to be able to establish itself in the host. Okay, so 3 different virulence factors: extracellular enzymes, and sometimes we refer to these as exoenzymes, same thing. Typically, we do a whole lab on exoenzymes. We test organisms for enzymes that are secreted outside of the cell. in order to break down some nutrient, usually like a protein or starch maybe, and then to bring in those smaller bits to metabolize them. The extracellular enzymes that we're talking about here, again, are ones that are secreted outside of the cell in order to break down some host tissue. Okay, we're going to talk about some toxins, a couple of different examples there, and also some anti-phagocytic factors. So those are going to be virulence factors that help the pathogen evade the host defenses. All right, so we'll take those one at a time. All right, some examples of some extracellular enzymes. So again, secreted outside of the bacterial cell here, hyaluronidase and collagenase. Two examples, hyaluronic acid is kind of the glue that holds cells together. That was a little sloppy. All right, so some bacterial cells can secrete an enzyme that breaks down that, again, kind of stickiness between cells and allows the bacteria to invade deeper into deeper cell tissues. Collagenase, collagen is a structural protein, the chief structural protein. Let's see. Main structural protein in our bodies. Okay, so if a cell can again start to break down that protein, probably going to chop it up into its amino acids. It's going to be able to break through this collagen and again deeper into our host tissues. A couple other examples, at least from what the book gives you, coagulase and kinase. All right, you should recognize coagulase if you're in lab. That's an enzyme that we tested to see if Staphylococcus produced that. That's an enzyme, again, secreted by the cell that kind of helps the organism form a clot. Okay, so why would that be advantageous? Here's a clot here. The organism has kind of formed a clot around itself to kind of wall itself off from the host's immune cells. So white blood cells that would come and probably, phagocytize those organisms aren't going to be able to get to them. So the organism has some time to kind of reproduce and build up in numbers. When the time is right or when the nutrients run out, it produces another enzyme, kinase, to break out of the clot and then move out into other areas of the body. Okay, so that's coagulase and kinase. So they're just some examples. There are probably lots of other ones. Okay, toxins. All right, a lot of bacteria can make some kind of toxin that's going to harm the host or maybe trigger some host immune response that's not fruitful or good for the host in order to clear the infection. Two types of toxins. We've mentioned endotoxin before, so we get to review that a little bit, and also exotoxins. So endotoxins and exotoxins. Okay, you may have remembered back when we talked about the structure of the bacterial cell wall, we talked a little bit about exotoxin. Exotoxin is actually part of the lipopolysaccharide of the cell wall. So let me remind you, I'll come back to that slide in a second. Hopefully this looks kind of familiar from about a month ago when we talked about the cell wall of gram negatives. Okay, so part of that outer membrane of the cell wall is this lipopolysaccharide. So that LPS is made of lipid A, which is embedded right in that outer membrane, and then a couple of polysaccharides that kind of stick up from that. Okay, so lipid A, that is endotoxin. That's the same thing. All right. Yes, it's outside of the cell, but originally we thought it came from inside of the cell, so that's why it was named endotoxin. Okay, endotoxin is only released when the cell wall breaks down. So that's kind of obvious when you picture it here. It's just kind of hanging out there in the cell and not doing any damage until it's released from the cell wall. When does that happen? When the cell dies, the cell wall breaks down. To some extent, when the cell divides, you could get a little bit of a release of endotoxin. Okay, so I have that here, released when the cell wall is disrupted. And most all gram negatives have this. There's different varieties of endotoxin and different varieties of lipid A that cause, maybe more or less damage. But what effect does that have on the host? It actually has a pretty low toxicity, meaning in small amounts, it doesn't really cause much harm to the host. But if you get a lot of it, it can be enough to kill the host. Okay, it causes things like fever and inflammation. I'm going to talk much more about inflammation as we get into immunology, but inflammation, it always seems like it's bad. Like we associate inflammation with pain and swelling. In small amounts, that actually helps our immune cells get to where they're supposed to go. But when that gets out of control, when our blood vessels get too leaky, blood pressure can go way down, host can go into shock. So too much inflammation is a bad thing. Our body tries to dial in what's right. But if you get too much of this endotoxin and too much inflammation, that can be deadly. Exotoxins then, so endotoxins is lipid A. Exotoxins are proteins. So hopefully you're thinking, well, extracellular enzymes are proteins too. What's the difference between an extracellular enzyme and a toxin? Exotoxins work systemically, so over the whole body. Okay, let's see. It only takes a little tiny bit of them. So they work in very small concentrations. And again, they can have systemic wide effects on our body. Extracellular enzymes only work right around the cells where they're secreted. Okay, they don't get into the bloodstream and cause clotting, for instance, all over the body. They just only work right where the cell is. Okay, so they're proteins. We'll say work systemically. Okay. They're typically very specific. In other words, they don't have these general effects, like inflammation. They work on a particular kind of cell, usually. So we'll see some examples of that in a second. They have very high toxicity. Okay, so that's related to the working in very small concentration. A 6-ounce bottle of botulism toxin is enough to kill everybody on the planet. Okay, so that's the most poisonous substance known to man. It's astonishing that we can use botulism toxin clinically to say, get rid of wrinkles or to calm muscle spasms. It has to be diluted millions of times in order to be able to not harm us and actually maybe work for good in some cases. Okay. All right. So if a patient has some kind of disease manifestation from ingesting a toxin, we would call that toxemia. Okay, so in some infections, it's not really the bacteria that harms us so much. Maybe we can give an antibiotic to kill the bacteria, but once they've produced a lot of toxin, that takes a lot longer to clear the toxin from our bodies. Okay, so something like Trying to think of an example. The whooping cough, that organism, Bordetella pertussis, produces a lot of toxins that it just takes a long time to clear those. And sometimes we can give the patient some kind of antibody that's going to neutralize the toxin a little bit. Okay, so here's some examples of exotoxins. A cytotoxin it's a very general term for some kind of toxin that would kill a host cell. Okay, again, pretty general term. A neurotoxin interferes with nerve cell function. So botulism toxin, tetanus toxin, those are so deadly because they interfere with nerve impulses. Enterotoxin, there's that prefix entero again. So that gives you a clue. Those are toxins that are going to target the intestinal lining, gastrointestinal tract. Sounds like endotoxin, different. Okay, different prefix there. Okay, so let's see, cytotoxin, a little comparison of exotoxin and endotoxin. And here I thought this table was pretty nice that the book gives you comparing endotoxins and exotoxins. We talked about most of this. Of course, endotoxin is only gram negatives, where exotoxins can be gram positives or gram negatives. Mostly bacteria we're talking about that secrete toxins. I don't know of any protozoa that can do that, but I could be wrong. Where it comes from, proteins, lipids, toxicity, we talked about that. Low, but can potentially be fatal. Don't worry about heat stability. We didn't really talk about that. What kind of effects there are in the host, we gave some examples there, whether it makes fever or not. This is sort of interesting as we eventually will move into our discussion on vaccines. Does antigenicity, that means does it produce an immune response in our bodies? So the exotoxins have a really strong immune response, so we can actually take a toxin and maybe add formalin to it or something just to change it a little bit so that it's not going to cause those bad effects in our body, but will elicit the production of antibodies against the real thing. Okay, so that's when you get your tetanus shot, that's basically a toxoid vaccination that if you are then at some point down the road exposed to tetanus shot toxin, you'll have the antibodies to neutralize it. So we don't have the ability to do that with endotoxin. We don't have any good way to neutralize those. And in some infections like meningococcal meningitis, the endotoxin is way, way, way more powerful than what it normally is in other infections, and that's typically what kills you. So interestingly, you might have known that there is viral meningitis and bacterial meningitis. You would think maybe viral meningitis would be worse because we don't have good drugs to treat viruses, but actually the bacterial meningitis is much more deadly because of the production of the toxins. Viruses don't produce toxins. Okay, so just some examples of diseases there. Again, don't memorize those, just some examples. All right, and then our last kind of virulence factor for gaining access to the host was anti-phagocytic factors. So let's kind of review a little bit of what phagocytosis is. We probably, I don't know, maybe we talked about this at some point in another class. We're going to talk about it in more detail again a little bit later. But phagocytes, these are white blood cells, again, in our bodies that engulf and hopefully most of the time remove invading pathogens. Okay, most of the time they're successful, at least we hope they are. So basically, some white blood cell, like a macrophage, for instance, would be an example, would engulf a bacteria, extended pseudopods. Remember, our white blood cells don't have cell walls, so they're able to do that. They enclose the bacteria in a membrane, which we call a phagosome. And then our lysosomes would fuse with the phagosome, now called a phagolysosome. And remember what's in lysosomes, lots of digestive enzymes. So we'll talk a little bit more about that later. We actually sometimes can produce a little bit of bleach in our white blood cells, lots of different mechanisms. So usually the bacteria is killed and the wastes are exocytosed, okay? So the problem is some organisms have ways around this, ways to either not be engulfed in the first place or to actually be able to survive the onslaught of the lysosomes. Okay, so some examples of anti-phagocytic factors, several that would prevent engulfment in the first place. So one that we've talked about already is that a lot of organisms make some kind of capsule. Okay, that just helps them protect them a little bit from being engulfed if they have that capsule. Now, eventually, our immune system has ways around that capsule. We have different kind of non-specific things like complement that we'll talk about later. Eventually, when we get a specific immune system mounted, antibodies can help our phagocytes get rid or be able to attach to an encapsulated organism. So this will buy the bacteria some time, but eventually, hopefully, our immune system can overcome that. Leukocidin, that's a particular kind of toxin that some bacteria can secrete that actually kills white blood cells, pokes holes in their membranes. Okay, so makes their cells leaky. They can't make that proton gradient to make ATP. Okay, so that's an example of an antiphagocytic factor. And one more that I'll mention, even though it's, I'm not sure if strep pyogenes is the only organism that makes M protein. There might be a few other streps that do it as well. But I thought I'd mention that because it's pretty effective. So this M protein on the outside of streptococcus cells, basically it inhibits our phagocytes from being able to engulf that cell. I mentioned complement real briefly. That's a protein that typically sticks to the outside of cells, and then our pseudopods and phagocytes can attach that and recognize that complement and be able to engulf them. So with a cell with M protein, that complement can't bind. So we'll talk more about complement later, but in case you're interested, why M protein is anti-phagocytic, we'll talk a little bit more about that. Okay, so just some examples. There is some organisms that actually welcome being phagocytized, but then they have ways to prevent the digestion. Okay, they have ways to survive inside the phagocyte. So for example, Mycobacterium tuberculosis, that's the organism that causes TB. It actually targets macrophages and it grows and reproduces within that macrophage because it has ways to keep the lysosome from fusing with the phagosome. Okay, so that's where it wants to be. It's like a criminal hiding out in a police station. Okay, the very cells that are sent to kill it, actually it takes over those cells and grows and reproduces quite happily. All right, so, you know, we have defenses, bacteria have defense, kind of like an arms race. We go back and forth. Well, they have that, and then we have this to counteract that. Okay, so we have talked about ways that the bacteria cells establish an infection. We'll step back for a minute and think about our host response. So mostly that's what our whole immunology section is going to be about. If we think a minute about, well, what's going on with our body? How do we describe what's happening to us in terms of stages of infectious disease? So if we take time 0 here is when that pathogen has crossed the portal of entry, you know, whatever way that is, whether it's swallowed or some sexually transmitted disease or whatever. how can we describe what's going on in our body? Well, typically there's always going to be some kind of incubation period. So that's a time where the organism is slowly or maybe quickly reproducing and gaining in number. They usually have to get up to some kind of critical number before it starts doing damage in our bodies. Okay, so we're graphing here either the number of microorganisms or the intensity of signs and symptoms of the host. A lot of times those two things go hand in hand. How long is the incubation period? There's a huge difference depending on what pathogen it is. For influenza, it's usually about a day of incubation period. For coronavirus, we think it's more like 4 or 5 days on average, but can be as few as 2 days, as many as 14. We still have a lot to learn about that. Sometimes organisms are infectious even in the incubation phase. So that's before there's any symptoms in the host. It's a problem to stop the spread of those infections. Chickenpox has a long incubation period, about 2 weeks, and that's when the host is most infectious, when they don't have symptoms. So again, it's really hard to stop the spread of an infection like that. Once the host is starting to exhibit some symptoms, we would call that the prodromal period. So that might be a term that you haven't really heard before. Again, kind of vague symptoms. You wake up in the morning and your throat's a little scratchy. You kind of start, something's not quite right. Okay, this is why we're doing our daily screening to try to catch those prodromal symptoms of coronavirus, try to keep you from going out if you got a little headache or maybe a little cough, not too uncomfortable yet. Normally you would just go about your business, but again, we know coronavirus can be infectious even then. So mild symptoms there. The illness phase is when the signs and symptoms become most severe. So some people really never get to severe signs and symptoms for coronavirus. The difference there between signs and symptoms, signs are observed. And a lot of times I would use these terms kind of interchangeably, but if you want to use them correctly, signs are observed. So things like fever, rash, vomiting. Okay. And then a symptom, they're just felt. So pain, malaise, fatigue, achiness, all right? So there's a difference in the definition of those two terms. So for some infections, we get lots of signs and symptoms, and then, if that kills the host, that you never get beyond that. But assuming that our immune system starts to kind of win some battles, we start to see a decline in the signs and symptoms, probably less of those microbes that are causing it, and eventually convalescence back to normal health. Okay. All right. All right, so let's talk about portals of exit, because obviously a microbe doesn't want to get into the host and then never leave. It wants to infect the host and then be able to infect other hosts as well. So one general principle is that generally our portals of exit are going to be the same as a lot of the portals of entry, but typically that microbe is leaving in much larger numbers than what it came. So that infectious dose may have only been, 10 or 100 microbes, typically going to leave in the millions. Okay, so respiratory and salivary portals, coughing, sneezing, laughing, talking, breathing. Of course, those are the ones that we're focused on with coronavirus. That's why we have a mask on right now. I've seen a few times people take their mask off when they have to sneeze. And that's like, oh, these breaks. It's like, no, that's why you have a mask on. You're not going to catch your whole sneeze in your elbow. So that's kind of the whole point there. Rabies has a salivary portal of exit. So That virus end stage is in the salivary glands. People that have rabies or animals, their throat gets extremely sore, so sore that they can't swallow. And so that's the foaming of the mouth that you see with dogs, drooling with humans because it's so painful to swallow. So that's its modus operandi. Fecal exit, so intestinal pathogens create motility of bowel. Those are scary words there, resulting in diarrhea. Cholera is an example of an organism that's really good at this. I don't know if you've ever seen a picture of a cholera bed. Okay, so, refugee camps, commonly you have some natural disaster, you get a cholera epidemic. Cholera kills patients by causing massive diarrhea, so much that patients dehydrate, and that can happen very quickly in a day or two. So patients typically lay on cots like this. They're too weak to get up to go to the bathroom, so they just let the diarrhea drain out. It just sounds awful. But save lives by rehydration. So it's like a bag of saline there. We don't even really have to give antibiotics there. And actually, I forgot to mention that cholera is a gram-negative organism. Sometimes we don't want to give antibiotics to treat gram-negative infections because that releases lots of endotoxin all at once. So in some cases where it's important to save the life to kill the bacterium, we would give antibiotics, and then we would give some kind of anti-inflammatory drug like Tylenol or ibuprofen, just so the effects of the endotoxin are not as bad. Okay, so rehydrate for diarrheal disease. Another portal of exit, of course, vaginal discharge, any sexually transmitted infection there. Sometimes the urine in kind of rare cases. Of course, bleeding, HIV, AIDS is a good example there. Okay. All right, so once the organism leaves the host, it wants to ideally, fall in some environment where it's not going to die right away. Okay, so we call like where the organism kind of hangs out and stays viable, those are sites that we call reservoirs. Okay, so sources of infection. You could also think about this, there's a homework question that asks you about this, sources of contamination. Okay, so where are humans picking up these infections? Usually from some reservoir. Okay, and reservoirs can be animal, they can be human, or they can be non-living. Okay, an animal reservoir. So typically, I'll mention this, typically the animal is not harmed. Well, that's not strictly the case. So for example, a raccoon here has rabies. Eventually rabies will kill the raccoon. But a lot of times a true reservoir is one that it's just kind of hiding out in that organism and it's not harming it, but it's able to transmit that pathogen to us. Okay, so infections that have reservoirs in animals, we consider these zoonotic infections. And these are typically pretty severe for humans because these are pathogens that have evolved to kind of not harm the animal, and maybe they have had some kind of mutation that makes them now able to be transmitted to us. We don't have the defenses for it. So oftentimes zoonotic infections are pretty harmful to us. Lots of the new infections that we have that are emerging are coming from some animal source. A great book, maybe over Christmas, if you're interested in it, in emerging infections. It's called Spillover by David Quammen. This was written about 10 years ago. Very readable, especially if you have a little background in microbiology like you all do now. Very interesting chapter called Dinner at the Rat Farm. Talks about SARS-CoV-1, severe acute respiratory syndrome. Really fascinating, talks a lot about that, and talks about how there's the potential for coronavirus to be a real problem. So he was really on the ball there. But talks about lots of different animal reservoirs. Bats. Bats is a really common animal reservoir. If you weren't previously afraid of bats, you would be after you read this book, because there are so many viruses, especially that have their reservoir in bats. We think coronavirus has a reservoir in bats, maybe Ebola as well. But why bats? Well, they live communally. So thousands of them live together in a cave, like right on top of each other. So they pass infections back and forth really easily. And then, when humans come in contact with bats, either by their droppings in some case, or rarely by a bite probably. I think bats are eaten in some areas of the world, so that's when they can pass it on to humans. Okay, so Lyme disease is another zoonotic infection. It's maintained in these white-footed mice, and deer to some extent as well. Insects are not considered reservoirs usually. We have another name for insects. We're going to call them vectors. So we'll get to that in a minute. Okay, sometimes humans can be reservoirs. An example of that, typhoid. You may have heard about the case of Typhoid Mary. Okay, she was actually, we call a human that was a reservoir a carrier. Okay, she had been sick with typhoid, and sometimes typhoid gets into the gallbladder and infects the gallbladder. People recover from typhoid, but they're still shedding virus from their gallbladder. So she worked as a cook, early 1900s, and probably didn't wash her hands after she went to the bathroom very often. And again, worked as a cook and killed lots of people because she was passing typhoid to them. And they tracked her down, and she was, kind of combative, and ended up living in quarantine for the rest of her life. So in rare cases, humans can be carriers. Of course, there's lots of things that we pass around to each other, colds, flu, that sort of thing. Here's another example that your book mentions of an example of a human carrier. Again, passing typhoid, early, I think it was 20s or 30s. It says there was a man who was a carrier for typhoid, and he habitually urinated at his job site, where they were laying new water pipes for a town's water system. And he just peed in the water pipes. I mean, that's sticking it to the man to a whole new level, peeing in your town's water supply. And lots of people got typhoid and died from him. Okay. Okay, some examples of non-living reservoirs. Food can be considered a reservoir. Soil or water. Okay, so any foodborne illness, even though that pathogen is probably maintained for a fairly short amount of time in that, potato salad or lunch meat or whatever, that could be considered a reservoir. Soil, in a few cases, for organisms that form spores. So botulism, tetanus, they can be, anthrax can be in the soil for long periods of time, maintained there. And water, there's some protozoa that cause diarrheal disease that live in water. For example, cholera actually can be maintained in water as well. So that's a pretty common one as well. Okay, so how do those pathogens get passed from the reservoir to us? All right, there's 3 different modes of transmission that we could describe. Of course, this is something that's endlessly debated with coronavirus, so we'll talk about that a little bit. Contact transmission. Okay, so that would include any kind of direct contact, like any kind of touching, like a handshake, kissing, sexual intercourse. Okay, all those direct kinds of touching, or some kind of indirect contact transmission by some kind of fomite. Okay, a fomite is an non-living or inanimate object. So an example of fomite transmission would be maybe a computer keyboard. I think we've pretty much closed computer labs on campus, haven't they? I haven't seen anybody in them. But if, say, somebody has the flu, they're typing away on that keyboard and maybe coughing on it or whatever, rubbing their nose, touching the keyboard. Somebody else comes in 2 hours later, that virus is going to stay viable for, whatever length of time. Person touches it, wipes their eye or whatever, bam, suddenly they're sick. So that would be an example of fomite transmission. Or droplet transmission. So this is the sneezing, coughing, maybe even talking. Droplets that are passed within 1 meter. So these are going to be fairly large droplets for the most part, if they are going to pretty much fall out of the air further than 1 meter from the person. Okay, so there's nothing magic about 1 meter. It's just sort of an arbitrary, point. If somebody sneezes into the air like this, an unstifled sneeze, you get really small microscopic droplets that can carry viruses and bacteria really effectively over much further distances. Okay, so our second kind of transmission, we'll call it vehicle transmission. Okay, that pathogen is hitching a ride on some kind of vehicle. It could be the air. Okay, so an airborne transmission, that pathogen is going to be on tiny, tiny droplets. They're going to float greater than 1 meter away from whatever reservoir it was. All right, so we think mostly coronavirus is by this droplet transmission, but there's evidence that it can travel further than 1 meter, certainly. So, unfortunately. Waterborne or foodborne vehicles, these could also be considered reservoirs, whether you consider them vehicles of transmission or true reservoirs, probably kind of depends how long the microbe is spending in that water or food. Okay. And it depends on the pathogen. Usually the food has not been cooked thoroughly and they've survived the cooking process. Okay, our last mode of transmission is by vector. So lots of vocabulary here. We know what a fomite, know what a vector, know what a reservoir is. Vectors are living organisms. I couldn't think of an example where that vector is not an insect. Okay, so it's usually some kind of insect that is transferring the pathogen from the reservoir to us, to the host. Okay, that vector can be considered biological or mechanical. Remember, they're both living. Mechanical makes it sound like it's a robot or something. But a biological vector, the microbe is growing and developing, so part of its life cycle within that insect. Okay. And a mechanical vector would just be a passive carrier. So you'd really have to know something about the pathogen itself or the infection to understand if that mosquito would be considered a biological vector or a mechanical vector. Okay, so if you take the instance of malaria, we talked a little bit about malaria. You know that part of the life cycle of that Plasmodium protozoa takes place within the mosquito. Okay, so that would be considered a biological vector. Most often that's the case. Bubonic plague, growing and dividing in the flea itself, that's part of its life cycles taking place there. When fleas bite us, then that's when we get bubonic plague. A passive carrier, the best example I could think of that is maybe a fly that lands on some manure, picks up some kind of fecal pathogen, and then lands on the potato salad at the picnic and just passes it on. Okay, so that microbe was just kind of hitching a ride on the fly. It didn't, really increase in numbers or, have any cysts phase of its life cycle or whatever. Okay. All right, questions. All right, we're going to finish up here with some terminology associated with epidemiology. So it's a hot topic lately, of course. Epidemiology, although it sounds really exciting, really involves a lot of number crunching, statistics, legwork, asking the right questions to try to figure out how diseases are moving through populations of people. So some terminology that epidemiologists like to use, they like to talk about the incidence and the prevalence. Okay, so know the difference between those. Incidence is the number of new cases in a given time. So it may be a week, it could be a year, depending. And then the prevalence is the total number of cases in that same given time. So the graph that your book shows you to illustrate this can be a little bit confusing because the scale is different for incidence and prevalence. Okay, so this is actually somehow my label got lost here. This is a graphical representation of HIV/AIDS incidence and prevalence. So back in the maybe 1993, it looks like the incidence of HIV peaked. Okay, so the new number of cases in that year looks like about 80,000. The prevalence, remember, that's the total number of cases. This is a little confusing because here it looks like the incidence is higher than the prevalence, but this is going by a different scale. So the prevalence here is more like maybe 150,000, something like that. So the prevalence is always going to be higher than the incidence unless patients die right away or they get better right away. Then maybe they could be about the same, but you would never have a lower prevalence than incidence. Okay, and then you can see, over time, incidence of HIV was going down. We're in better education. A little uptick here. I don't have the data any more recent than that, but I don't think that that's been going higher. Prevalence, on the other hand, is skyrocketing. It's because people are living a long time with AIDS. So even though the new cases are going down, there's a lot more people in the population that have HIV, or just living with it. Okay. Some other terms epidemiologists use. Again, some of my words get put on top of each other. But you should know the difference between these terms: endemic, epidemic, sporadic. An endemic infection is one that's constantly present in a population. So colds, flu, we always have those things with us. They're kind of, seasonally going up and down. If it's sporadic, you might have a few cases here or there, but not, like one case doesn't seem to cause a big cluster. The term outbreak could be on here as well. Whether you call something an outbreak or an epidemic, both those terms describe an unusually large number of cases. I think an epidemic would probably be over maybe a wider area, where an outbreak could be like, we had an outbreak of Eastern equine encephalitis in the fall of 2019. But maybe sometimes you would call that epidemic. We had so many cases. So those two terms are a little bit blurry between them. A pandemic is when an epidemic is on more than one continent. All right, so how do you know if a disease is epidemic or not? Well, you have to know the baseline. So there are tables, there are notifiable diseases that you can look up. So I just did a screenshot of this for an example. If you go on the CDC website, you have to kind of hunt for this a little bit, but there are certain diseases that if you go to the doctor and you get diagnosed with, your doctor has to notify the CDC. So things like malaria, you know, all of these infections here, cholera. Coronavirus is not on this list, but it probably at some point will be added to it. Okay, so you click on whatever disease that you're interested in. And then, for instance, I clicked on measles and malaria. And so this is the data for just a couple of weeks ago. And I thought this was kind of interesting because last year, in 2019, there was almost about 1,000 cases of measles in the US. So that was a lot. This year so far, we have 7. So we're doing a good job of not passing measles around this year. And you could look at individual states. So it's kind of interesting, like what we normally have, so you can understand if, oh, is this a lot or is it just kind of normal? Okay, here's malaria. Last year we had a lot more malaria too. We're traveling less. So of course, malaria is not endemic in the US. Anybody that has it would have brought it in from another place. So it's kind of interesting to look at those numbers. And here's another just graphical representation of the difference between those terms. An endemic disease, it's just kind of normally present, sporadic here and there. Something like, I don't know, botulism. So you get people here or there that don't can their vegetables correctly. They might get botulism, but they're not giving it to a bunch of other people. So it's not really an outbreak. A large number of cases in one place, an epidemic. And one more graph here. Again, just understanding epidemic versus endemic. This disease here kind of goes through seasonal fluctuations, but the actual numbers are pretty close to what we'd normally see. So that's endemic. If you have a big spike in this other infection here, then you could consider those spikes an epidemic. Okay. Are there questions? Okay, so that is the end of our material for exam 3. Okay, I have enough time here that I think I'll at least start our introduction to our next material. I guess I'll just kind of finish up by saying here, several of you have asked me, I just don't know how to study for this stuff coming up here for the exam on Tuesday. For the antibiotics, focus on those mechanisms of action. So I know we did, what was like 14 or 15 different drugs, know how those work. That's probably the most important thing you can learn if you have to learn one thing about the antibiotics. Do you have to know every little thing, like whether they're acid label or how they're administered? No, but you should understand those principles. Like, what does it mean to be acid label? What are the different administration methods and why we might use them? Question, Ashley.
Are you going to post a small practice questions?
Am I going to post practice questions? Yes, I have those ready. I'll post them after lecture because I know I haven't been really giving you very many as we go along here. I want to make sure I got through the material. So I have like 7 or 8 that I'm ready to post. Okay, and they're from previous exams. The antimicrobial methods, I would just kind of go through each one and think to yourself, okay, what do I know about this? When would we use this radiation, for example, or how does radiation kill cells? We talked about both ionizing and non-ionizing radiation. Common uses. anything just sort of, interesting, like for the antibiotics, rifampin turns your urine orange. Okay, that's kind of striking. Chapter 14 here, like I said, has been a lot of terminology. So any term that you didn't recognize, make sure you understand how that's used. Any other questions, I guess? I posted those objectives, look through those, yeah.
The antibiotics, you said not to be worried about specificity for like acid labile, but what about like administration techniques?
Yeah, so what about the administration techniques for antibiotics? No, I don't think so. Mostly just understand if the therapeutic index is low, then we're going to want to administer it by some kind of injection or IV rather than orally.
Okay.
So that's
rather than like seeing that like penicillin G is usually injected kind of stuff. Like don't worry.
right. So don't worry that we would inject penicillin G, right? All right, so I'd like to do a little introduction to our immunology. We're going to be spending several weeks on immunology. This will take us up to Thanksgiving. These are going to be chapters 15 and 16 in your book. And I would really encourage you after Tuesday to actually crack your book if you haven't done so yet and read these chapters. They are kind of long chapters, but we're covering most everything in them. And mostly what we talk about in lecture, you're probably, if this is all new to you, you're probably going to need to see it a couple different times till it sinks in. So reading your book, hearing about it, maybe just said a little different way. There's going to be lots of animations in the study helps in mastering. The homework is kind of extensive for both chapter 15 and 16. So kind of plan on throwing yourself into that a little bit in order to do well. What is immunology? Basically, it's the study of how our body protects us from all those pathogens that are trying to establish themselves in us. So viruses, bacteria, fungi, protozoa, even parasitic worms. There's lots of stuff out there that's trying to invade us. And most of the time, we don't even know, that our immune system has fought something off. Our immune system has other functions. I know I didn't post this slide, just repairing wounds. There's immunological things that has to happen for tissue regeneration, tissue cleanup. So our cells wear out. We have macrophages that go around and eat up, dead cells in our body. Okay. Unfortunately, our immune system has the potential to cause damage itself. So if something's not working correctly, that's when we get autoimmune disease. Okay, and then we have to, in most cases, take some kind of a drug to dampen our immune system. Of course, that's not good. Then we're a little bit more likely to get some kind of infection. So we'll talk a little bit about, when that might happen. I definitely didn't want to post this because I didn't want to scare you, but immunologists are really fond of posting diagrams, really complicated diagrams like this. Okay, immunology is a fairly young science. We're really learning a lot about the function of our immune cells. So that's one thing that makes it kind of difficult to learn about. The other difficulty is that, say we talk about macrophages. Well, I can't really talk about macrophages without talking about the effect that they have with B cells and T cells and how they affect with other kinds of cells. So everything's sort of interconnected. So we have to sometimes I have to mention something and say, well, I'm going to talk about that more later, but for now, this is what you need to know about it. So it's good for a healthy immune system that we've got overlapping function. All right, if say a person's neutrophils are lacking, other cells kind of take over and fill that void. Lots of different signals here that happen between immune cells. We call those cytokines. And for the most part, we're not going to worry about what cytokines do what, thankfully. We're just going to say, oh, there is some cytokine messenger here, and that's how one cell can kind of impact another. All right, so this is the way that your book kind of is going to come at the immune system. I think it's a pretty good method to look at it from kind of a first line of defense, second line of defense, and third line of defense. Okay, the first line would be kind of just in a nutshell here. This is our external barriers. So our skin, that's a really good first line of defense. We've got, I mentioned today, we've got defenses at our mucosal membranes. I'm going to talk again about how the role our own bacteria plays that's normally there in fighting off other kinds of infections, some kind of antimicrobial peptides. Okay, that's all considered part of our innate system. So innate means that every time our body encounters a pathogen, basically it has the exact same response. And that's a little bit of a simplification, but in general, that's the case. So same response. Same thing with our second line of defense, which mostly includes cells, like our white blood cells, some non-specific things like complement proteins, interferons are a cytokine that helps us fight off viral infections. I mentioned today that inflammation usually is good if it's in the right amounts. Even fever can be good to help us perhaps fight off an infection again. But those are all responses, again, that are pretty much the same every time our body encounters them. And then our third line of defense is our adaptive immunity. So that involves our B and T cells and how they fashion an extremely specific response in order to either kill a cell that's been infected with a virus, that's our cell-mediated immunity, or humoral immunity that involves the production of antibodies. All right, so that's a little overview of our immune system. Let me ask you this question. I don't know if you ever played the game, Would You Rather? So that's like, would you rather eat a toad or a snake? Those sorts of questions, like neither one. Okay, if you had to do without one branch of your immune system, which one would you choose? Your innate immunity or your adaptive immunity? And it turns out there is a good answer to this question. But think about that. Which would you pick to do without? Okay. Usually I have students like answer this, and the clicker question is usually about 50-50. Okay. Well, some interesting experiments have been done that deal with this question. Okay, it turns out that if you had to choose one to do without, you would probably want to do without your adaptive immunity. Okay, if you knew that some mammals and birds don't have an adaptive immunity, then that might have helped you answer that question. But here's the reasoning behind that. So we can all do all sorts of crazy things with mice now. Knockout mice, we can have mice that are able to do certain things or not able to produce certain cells. So here's a norm. You can look at the normal graph here first. So humans, normally when we're fighting off an infection, the number of microorganisms rises and then our immune system fights it off. If we were lacking adaptive immunity, we really, we would have some defense, but usually not enough to totally resolve the infection. But if we're lacking innate immunity, totally have very little protection at all. And the way that we figured that out is we took some mice, they're called SCID mice. So that's severe combined immune deficiency. I guess these are mice that have polymorphonuclear leukocytes. That's a kind of white blood cell. And they have macrophages, but they are not able to make T cells and B cells. So they have these really important cells of our innate immunity, but they are not able to make these adaptive immune cells. Okay, so again, they have some defense with the PMNs and the macrophages, but then they can't totally get rid of the infection because they can't make antibodies. Okay, and here's mice up here that are lacking innate immunity. They're lacking some of these non-specific kind of first responder types of cells. And again, that the infection pretty much goes unchecked. So, you know, the correct answer is we want to have both aspects of our immune system. And there are people that are born with genetic deficiencies in one or the other, and it's a rough go. Okay. I think that's probably a good place to stop with our introduction. We'll start in, even though I'm giving you a 5-minute break here. We'll start in with our first line of defense on Thursday. I have office hours now, office hours by Zoom tomorrow. So come see me if you have any questions. Have a good day, good weekend.